Just a quick note, there's a little confusion. I'm actually don't have the doctor title yet. I am a graduate student here at the Andel Research Institute. My name is Alysa Kasem, and I work with Dr. Mike Henderson here. And again, just as a reminder, if I use language or terminology you don't understand, just interrupt my talk and we can talk about it before we move forward so everyone's on the same page. And today I'm going to talk about pathology and the impact on Parkinson's disease, which is a little bit different than what most people discuss. So just quickly as an overview of my talk today, I'll briefly talk about the pathologies we see in Parkinson's disease, specifically, then move into what the tau protein is and what it does, how it aggregates in Parkinson's disease through a few different aspects, like genetics, the prevalence of these aggregates, and a few current hypotheses on how tau aggregates in disease. And finally, how we can measure this tau aggregation in patients. So just briefly, as I know we've been hearing already today, but alpha-synuclein is really the main pathological protein in Parkinson's disease. And that just means it's the protein that aggregates and is associated with disease. And it forms these red Lewy bodies you can see inside this neuron. And just as a quick little lesson on neurons too, this part of the cell is called the cell body. And this is the axon or projection of the neuron. And that'll just be relevant for some images I'll show you later in the talk. So we know that alpha-synuclein aggregates and causes the dopamine cell death in the midbrain and substantia nigra specifically. And that's where we see the onset of those motor symptoms. But what's less talked about and is becoming an active area of research is actually the spread of these alpha-synuclein aggregates out of where the substantia nigra is in the midbrain here, out into the outer regions of the brain and the cortex. And what's been found recently is that the presence of those alpha-synuclein aggregates in the cortex and cell death there is actually correlated to onset of dementia-like symptoms or cognitive decline in disease. And when we're discussing cognitive decline in Parkinson's disease, it's really important to understand a few key components of it. And first, that's alpha-synuclein pathology, so that aggregated protein. The burden of that and cell death correlate to the risk of cognitive decline. There's also been studies that show up to 80% of Parkinson's disease patients that have been living with disease for over 10 years do experience mild cognitive impairment, and this just increases as time continues on as well. And that Parkinson's disease patients are actually 4 times as likely to develop dementia or mild cognitive impairment compared to the general population. So this is a really interesting finding because it makes us question whether there's something occurring in Parkinson's disease that is a similar way to what happens in dementia and Alzheimer's. And when we're actually trying to study this, if we look at a Parkinson's disease dementia brain, this is on the outer cortex region, when we stain this tissue, we can see aggregated alpha-synuclein shown in blue. And this is a Lewy body here, that big aggregate, and then we also have Lewy dendrites, and that's small amounts of aggregated alpha-synuclein in the projections or the axons of the neuron. And when we look at the same piece of tissue, but at a different pathological protein, a different aggregated protein, we find aggregated tau. And this is really interesting to us because this tau, in some cases, occurs in the same cells as the alpha-synuclein pathology, as in here, or in neighboring cells, as you can see them near each other in other places. And a study in 2017 found that nearly 90% of patients with Parkinson's disease or Parkinson's disease dementia after passing actually have this aggregated tau in their brains. And so it really makes us question what is happening here to make tau aggregate when we know alpha-synuclein is that main protein. And this is really exciting to us when we think in the context of dementia onset that we see in late stages of disease, because tau is actually also associated with Alzheimer's. It's one of the two main pathological proteins there too. So we have tau and amyloid beta in Alzheimer's disease. And what's really important to note from this image in this graph from a publication is that the increase in tau pathology that you can see by this red bar here is more correlated to the actual onset of the mild cognitive impairment symptoms and then the worsening of that cognitive decline over time. So as the tau pathology increases in the brain, we see worsening dementia symptoms in these patients. And so that makes us wonder if the tau pathology we are starting to see in the brains of Parkinson's disease patients is also linked to dementia symptoms. And so when we're talking about tau, I really want to talk about what this protein is and what it does. So tau is this smaller protein. It's coded by the MAPT gene, and that's just part of a gene in your sequence or in your genome that gives the cell instructions to make the protein out of those amino acid building blocks. And what we see is in neurons, if we zoom in on this region here, we see these blue structures called microtubules. And these you can think of like the highway and bridges of the cell. These are really important because they provide structure, but they also allow the movement of different proteins or different parts of the cell to move from one end to the other. So here, you can think of it in this cartoon schematic of a microtubule bridge with a little car. And the cars of the cell are called vesicles, and they can move different parts of the cell from one end to the other along these microtubules. So if we zoom in on these microtubules further, what we actually see is the protein I'm showing in red is tau. And tau binds to these microtubules and performs a really important function here. And they keep the microtubules stable. They're really important in keeping that structure together. They also control how these microtubules interact with each other. So if you think about how like roads cross, these microtubules can cross too. And tau really controls that. And it also controls what these vesicles or these vehicles of the cell are moving along from one end to the other as well. So now that we know one of the biggest functions of tau, it's important to know how this is important in disease. And what happens in tau aggregation is that the tau will come off of the microtubules, and this is for reasons we really don't understand at this point. We just know it comes off the microtubules, and it can clump up and form those large aggregates similar to what we see with alpha-synuclein. And when the tau falls off the microtubules lose their stability and can actually degrade. So we're losing the structure of the cell, and we're losing the ability to move different cell components from one end to the other. And this can make the neuron very unhealthy and can contribute to cell death. And so this leaves this question of how tau actually becomes aggregated. We don't know the why, but we know how it works. And this is very similar to what happens with alpha-synuclein that Saima and Raz already spoke about today. So we have a normal tau protein, and this is a cartoon version, but it looks pretty similar to this when it folds up. One end just interacts with the other, almost like a little paperclip. And normally in the cell, this protein can become modified. I'm showing that by these little yellow dots. And you can think of the modification similar to if you're following a recipe and you add seasonings or spices to it, right? And it can help your recipe come together. It helps the protein keep its shape and function well. But what happens in disease is that the protein gets too many of these modifications. So you way oversalt your food and it's no longer edible. I know we've all made that mistake. That happens here too, where the protein gets too many of these modifications and it makes it fold wrong. And like we've already talked about, when a protein loses its really important shape and structure, it loses its function. And when we have a lot of this highly modified, misfolded protein, it can form clumps and aggregates. And this is what we see happen in disease. Specifically, this is well characterized in Alzheimer's disease. And these clumps form these large inclusions and aggregates within the neuron, and it can contribute to cell death, just like it does with alpha-synuclein. So now that we know how it misfolds, how it aggregates, it's important to understand how this works in the context of Parkinson's disease, because that's why we're all here. And when we look at the genetics of Parkinson's disease, right, we have genetic variants. You have genetic variants all across your genome, right? And that's just a change in the genetic sequence in your DNA. Some of these variants don't do anything. They aren't associated with any disease and they're normal and they make me look different than Helen here, right? But some of these variants will either increase your risk for disease or can even decrease your risk for developing disease. But the risk is the key term here. Most variants don't directly cause the disease, but it's going to give you increased risk. So when we look at the genetics of Parkinson's disease, which have been heavily studied, what we see up in pink here in this top corner are very, very rare variants of different genes that are thought to be causal for PD. So these are greatly increase your risk or almost associated fully with having the mutation or variant and getting the disease later in life. But what's more common, what we see more often, actually variants that are more common in the population, but they only slightly increase or decrease risk of disease. And that's where we find the MAPT gene, which is the gene that encodes for that tau protein. So there is some genetic link for tau function in Parkinson's. But since we know there's a genetic link, this doesn't happen in every patient. But from what I've said earlier is we see tau aggregates in a majority of patients at some point over the course of the disease. So we really want to understand how prevalent these are. And I have quite a few pictures in the next slides of brain tissue that was generously donated by patients upon their passing. So I'd just like to take just a second and state that and just again thank them for their selfless donation, because that is really helping drive our research here. And what we see that if we look in the brain of a Parkinson's disease patient, and here we're looking at the amygdala. Oh, let me move this. Oh, I can't move that. There's a small blue region you'll be able to see in a minute with an arrow pointing to it. There we are. And that's the brain region I'm looking at here. It's called the amygdala. And when we look at this brain region in Parkinson's disease, we see in purple the what's called phosphorylated alpha-synuclein, and this is just a marker for that aggregated protein. We see these large Lewy bodies here, and we also see Lewy neurites that I talked about earlier, and those projections of the neurons. But in the same tissue, again, we see phosphorylated tau, or that aggregated tau, and the phosphorylation is just one of those modifications I mentioned earlier. So we see this aggregated tau in Parkinson's disease patients, but again, we also see this in Parkinson's disease dementia. Again, the colors are the same, where we have these large alpha-synuclein aggregates called Lewy bodies down here, and we have a lot more aggregated tau with Parkinson's disease dementia patients. But this isn't localized to just one region of the brain. So here we're looking at the same patient. So I have tissue from the amygdala and the midbrain from this patient who selflessly gave us their brain to study. That again, in this other region that's near the amygdala, but is just down here, we see again, we have alpha-synuclein aggregation, Lewy bodies, which is starting like in this cell here, but we also have aggregated tau in that region. And this really makes us wonder how important this is for progression of disease, right? It's not alpha-synuclein, it's not causing motor symptoms, but maybe this has something to do with the non-motor, the cognitive symptoms that we see. So why should we even study tau aggregation in the context of Parkinson's disease? And again, this is because we see tau aggregation correlating to worsening cognitive decline in Alzheimer's. So a different disease, but the same protein aggregates. And what you can see in this graph here is that when we have a cognitively unimpaired patient, they measured the levels of aggregated tau in the blood and blood plasma, which is a component of yours. And as cognitive impairment worsened, so as the patients progressed throughout disease, what they found is that the levels of aggregated tau in the blood also increased. And when they looked at then trying to understand the relationship of the amount of aggregated tau in the blood and how that might correlate or correspond to worsening of cognitive symptoms, what they found is that as plasma levels of the aggregated tau increase, so as we go to the right on the graph here, the MMSE, which is the mini mental state exam, score decreased. And a decrease in that score is worsening of cognitive abilities. So this really leads us to ask this question of, we know this is correlated in Alzheimer's disease, but is it correlated in Parkinson's disease? And is this tau pathology a coincidence? Or is there something going on in the cells that are linking this alpha-synuclein aggregation to tau aggregation? And what we're able to do in the lab is ask these questions in very simplified model systems. So one of the things we're able to do is grow neurons, or those cells that are in the brain, in small dishes. Raz mentioned earlier, a plate about this big can hold 96 wells, and we're able to grow neurons, because they're very, very small, in all of those wells. Around 200,000 neurons in each little well, and they're about the size of an erase rocket, the well is. So these are very small cells. And what we're able to do in this really simple system is then add aggregated alpha-synuclein to the symptom. We put it through a process that makes these aggregates very, very small, and add it to where these neurons, add it to the plate of the neurons. And this is a response we like to watch and understand, because what has been shown is that if we have the aggregated alpha-synuclein and we put it in a system where we have a normal protein, the endogenous alpha-synuclein, that's normal, it performs its function how it should, the toxic state or the aggregated state will actually cause the normal protein to become aggregated and become toxic. And you think of this like when you buy strawberries at the grocery store, they all might look really nice, but if one strawberry gets moldy, it spreads to all of them very quickly. It spreads that toxic state, just like these proteins do when we're talking about protein aggregation. So when we look at these cells, and And these are neurons, and I'd just like to thank one of the research team members in the Henderson lab, Libby, who so kindly did this work for me over the past couple months. She grew neurons in these dishes, and you can see we have these round circles that are the cell bodies here and here. They're all over. And we have the lines that are the actual projections of the neurons in these wells. And when we treat these with the alpha-synuclein fibrils and let them just stay and grow for 2 weeks, we then are able to take these cells and stain them to look for the presence of the aggregated alpha-synuclein and here aggregated tau too. And what we see is quite drastically is these cells have this toxic form of alpha-synuclein. So they took it up from the environment, and now it caused the normal alpha-synuclein protein to become toxic in these cells. And that's shown in purple. And you can see all these aggregates throughout the cells, both within the cell bodies. That would be similar to what we would call a Lewy body, and then in the axons, like a Lewy neuronate. But what we also see when we just add the alpha-synuclein aggregates, we also see tau become aggregated and toxic, which is pretty unexpected and not something we ever considered happening, because we wouldn't expect tau to respond to alpha-synuclein. But this leads us to ask this question of, how does this happen? Because we know it happens now, and we, with this data and this evidence here, and so many publications that have now done this as well, we know that this alpha-synuclein aggregation we see in Parkinson's disease might be causing tau to aggregate.
Could you repeat that, please? I didn't hear.
Yeah.
Okay.
So now that we understand that when we sorry, now I gotta think about what I said. We know that tau can become toxic in response to just alpha-synuclein, and we know that we see this in patients too, where we see aggregated alpha-synuclein and aggregated tau together. So now that this has been shown by a lot of different studies in a lot of labs all around the world, that this tau can aggregate, normal tau can aggregate with alpha-synuclein, we really want to understand what mechanism is happening for tau to become toxic when we have toxic alpha-synuclein. Does that make sense?
I could hear it that
Okay. Yeah, sorry. I do speak a little quieter, so if you can't hear me at any time, please just let me know. And there's a few thoughts currently in the field on why tau might aggregate in Parkinson's disease. And the first one has been termed the mitochondrial dysfunction hypothesis. So in a cell, simplified cell, we always have mitochondria. And the mitochondria take glucose, or this is sugar, and turn it into energy for the cell to use, just to perform its normal tasks, to make proteins, or for a neuron to work so we can think and move and do all these things. And what's very interesting about neurons in the brain is, even though the brain only accounts for 2% of your body weight, just when you're sitting down like all of you are now, your brain is taking 20 to 25% of all the glucose you have in your body to function compared to all the other organs. So that's quite a drastic need the brain has for glucose, and a need for these mitochondria.
Does that mean we can all eat lots of chocolate?
Yes. I am a big fan of eating a lot of chocolate. So. So the brain really, and these neurons that we have, really rely on the mitochondria to function. So that's part of why we think that maybe there's something might be happening to cause the mitochondria not to function right and maybe harming the cells and maybe leading to them to become damaged or die in disease. So this is a pretty complex pathway, but I'm going to try and break this down because I think it's important to understand how we think about this disease in terms of the research we do. So here we have just a mitochondria. It's split in half. It usually looks like a little kidney bean. And when a cell has aggregated alpha-synuclein in it, so this is a neuron that already has the aggregated protein, this aggregated synuclein can actually physically come in contact with the mitochondria. And the mitochondria do not respond well to this. They get very stressed. It causes physical damage to the mitochondria. And the mitochondria send out these specialized signals to the rest of the cell. And here I'm just going to call them just damage signals for sake of clarity. And this tells the rest of the cell that, hey, I'm damaged, something weird is going on over here. But when these damage signals become too much or too close to the microtubules that I talked about in the beginning, it can actually damage them. So when there's too much of these damage signals in the cell, we harm the microtubules to the point that they start degrading and the tau falls off that we talked about in the beginning. So when this tau falls off, there's a long pathway that happens, but in short, this tau gets dissociated or just free floating, not doing its function anymore, can turn into tau fibrils in the cell. And like alpha-synuclein, these tau fibrils can go and physically come in contact with more mitochondria. And there's a lot of mitochondria in neurons. Some estimates think up to between a few 100,000 to up to 1 million mitochondria are in a single neuron. So there's a lot of mitochondria for these fibrils to come in contact with. This causes the mitochondria to release more damage signals, causing more microtubule damage, causing more tau to fall off, and leading to the formation of more aggregated tau fibrils. There's another pathway too that happens, it's thought to happen with just this original microtubule damage too though. When the microtubules start falling apart, the cell recognizes that something's not right. It starts sending out more damage signals to try and recruit specialized proteins in the cell that are supposed to fix cell damage. So more damage signals are sent out. And what's interesting here is that alpha-synuclein, we don't really know why, but when the cell sends out these damage signals, it can actually make the cell produce more normal alpha-synuclein protein. It's thought that maybe this has something to do with fixing problems or maybe cell stress or even inflammation in the cell. So we have more normal alpha-synuclein. And what I told you earlier was that the aggregated form of alpha-synuclein can interact with the normal form and make it become aggregated. And that's what we think happens here, where this increase in normal alpha-synuclein protein causes more fibrils to form. Those fibrils interact with more mitochondria and increases stress and presence of fibrils in the cell. And now this is a cell that already has alpha-synuclein fibrils, but this is really important because if you were at Virginia's talk this morning, she told you there's a lot of evidence to suggest that these aggregations can spread from cell to cell. So if we're having a lot of alpha-synuclein pathology or even tau aggregates in these neurons, it can spread to neighboring cells. And this is thought to maybe be a reason we see alpha-synuclein spreading across the brain in Parkinson's cells and even tau here. And we can see this in our model systems that we study in the lab on a regular basis, but What's more important is trying to see if we can measure this in patients, right? Because we want to make sure our models that we have, or these tools we use to study Parkinson's disease, are representing what we see in patients, so we can really understand and treat Parkinson's. And I don't have any exciting news on this front right now, but I do want to say that there is a really great study that's going on right now through the Michael J. Fox Foundation. And this is in Pittsburgh, Pennsylvania. And these two researchers are looking at PET imaging. And this is a special type of imaging in the brain of patients that were able to look at specific proteins or markers if we put in a specific tracer. And I'll talk more about that in a little bit and just how this all works to make this make more sense. But there is ongoing studies looking to see if we can understand how the mitochondria and how damage to the mitochondria might be affecting disease. And this isn't the only hypothesis about how tau aggregates. And it's also important to talk about inflammation. And you may have heard a lot of associations or talk of specific infections being important in Parkinson's disease. And just to go over what inflammation actually is, so we're all on the same page, is anywhere in your body, your immune cells might see something as foreign. Like think of a cold or a virus that you get, right? Your body's going to recognize it as this is not what should be here. And it's going to mount an immune response. And this is where you'll get like a fever. It's your body's immune response to a foreign object that it doesn't want there. So it mounts this big response. And then as time goes on, we see this response decrease, right? Your fever goes away, you start feeling better, and the virus or bacteria that was making you sick is gone. This inflammation process is pretty similar in the brain. Although we don't get a lot of viruses or pathogens in the brain like we do in the rest of our body, there are things that can cause inflammation. These can be like hitting your head hard. That can make these immune cells in the brain called microglia say, hey, something's wrong, we gotta fix what's wrong over in this area that got damaged. Or even different, there are specific toxins or viruses that can go in the brain. They're very uncommon, but they do exist. And normally, when we have inflammation anywhere in the body, and specifically in the brain, the cells can become damaged and some of them can even die because of the inflammation. And normally when we have small amounts of inflammation, this isn't a problem because there are so many neurons in your brain, they can cope with losing a few here and there. This becomes an issue though when we have what we call chronic inflammation. And that's this purple line here, where your body starts an immune response, or these microglia in your brain start an immune response, and then this immune response never decreases. It just stays elevated. And
Can I ask a question?
Yeah.
I'm sorry. Does inflammation mean swelling, or does inflammation have a different definition? Like,
yeah, so
does it
it
make
can,
sense?
it can mean both. So here, inflammation is your general immune response. Normally we see what we normally call swelling as inflammation because your body is increasing blood flow, so more white blood cells can go to that region and deal with the pathogen or virus. So that when we think of inflammation, it's called swelling a lot. That's what the immune response tends to do. So the actual swelling tends to be part of the general inflammation response.
So there could be no swelling, but you still have inflammation. It could be marked by something else. Or no, it's not.
I'm not
Oh, that's okay.
super well versed in
That's okay.
immunology, but they usually coincide together.
Okay.
Usually we do see increased blood flow to regions. In the brain, it's a little different because we don't have the white blood cells you have like in your arms, in your brain. They're separated from your brain is separated from the rest of your body in that respect. So we do have the specialized microglia that generally don't cause as much swelling, but they do have all of the cell signals that are sent out. And those are what's really damaging to the cells, the neurons. So in the case of chronic inflammation, we have these large amounts of signals in the brain that are saying, I'm damaged, to do something about this unhealthy state. And this can cause widespread neuron death or widespread neuron dysfunction. And that's where we see issues and potentially a link with inflammation in the brain and neurodegeneration. So what does this actually mean though in the context of proteins aggregating? And what we see is our microglia can start this inflammation response where your head got hit really hard, or there's some specialized virus in the brain, or there's maybe something else going on, a genetic variant that makes you at risk for getting inflammation in your brain. And what these microglia do is send special signals to the neurons that they are next to activate kinases. And kinases are another protein in the cell, and these add the modification to another protein. So these are just going to modify tau, or if we think back to our seasoning analogy, these are the chefs, right? These are going to add something to the protein. And normally that's okay if there's only acute inflammation. But here, let's talk about chronic inflammation. So there's a lot of activation, there's a lot of signals, and the kinases are being told, modify more proteins and keep modifying them. And that's where we see the over-modification of tau again, where it misfolds. This misfolding, again, as I talked about earlier, can lead to the tau clumping up and forming aggregates. And what's really interesting here is microglia can recognize both aggregated tau and aggregated alpha-synuclein as a foreign substance and create a new immune response to the aggregates. And so this just increases the cycle more. It creates more inflammation in the brain. And this is thought to potentially be a reason why we could see tau aggregation and even spread of alpha-synuclein aggregation in the brain. And there are ongoing studies now looking to connect inflammation in the brain, looking at these specialized signals that the microglia send out in CSF, in the cerebrospinal fluid. And those are ongoing, but I won't talk about any of them right now. Because I think what's more important is to talk about how we can measure this tau aggregation in Parkinson's disease patients. This is can be done in really 3 ways. Looking at plasma, which is a component of your blood, cerebrospinal fluid, the CSF that gets taken from that lumbar puncture, and PET scans. And that's a specialized brain imaging that we're able to do in patients. So first, just looking at plasma levels. So if we look at plasma, which is part of your blood, and this is a study that was measuring aggregated tau in Parkinson's disease patients. Well, they measured aggregated tau in a lot of different alpha-synucleinopathies, like dementia with Lewy bodies, MSA, but I just want to focus on the Parkinson's disease patient, which is what I've highlighted here. And you can see between healthy controls here and the Parkinson's disease patient, there was an increase in the amount of aggregated tau in the plasma. You can see that because these are higher up where the number is. So we see this increase in tau in the blood. And the study was actually replicated. Again, this original study was done in 2018. Another study was done in 2022 looking at this again in a different population of patients to see if this. You can see again, the Parkinson's disease patients do have higher aggregated tau in the plasma. But I think what's really nice about looking at these graphs is each dot is a patient. You can see there's a lot of overlap between the controls and the Parkinson's disease patients. And so when we're talking about biomarkers, it's really important that we create a panel, right? A panel of biomarkers where we're able to group patients and say, you have these 5 biomarkers at these levels. So this is the disease you think you're gonna have. And this is the risk of you developing cognitive decline later on. But what's really beneficial in the field of looking at tau aggregation is these have been really heavily studied in the context of Alzheimer's disease. So these assays and these tests, they're already developed. We just need the patient samples now and researchers to take these patient samples and measure the aggregated tau there. Because the hard part of developing these tests is done. We just now need to look at the patient. When we're talking about cerebral spinal fluid, this is a lot more understudied in Parkinson's disease. There's, I think, 2 studies that I was able to find that actually looked at this, and the results weren't very convincing. But again, these are well validated in Alzheimer's disease, these studies and these assays and tests. So it's really going to be taking all of the tests we can run and using the samples from Parkinson's disease patients, both in early disease and late disease, because it's really important when you're trying to develop biomarkers to understand what happens over the course of disease into these tests and trying to understand how aggregated tau changes over the course of Parkinson's disease so we can better understand and treat the disease as well. But I do want to show this, and this is in Alzheimer's disease. So again, it's different, but we're able to just appreciate what these assays are able to do and these tests are able to do as they used 3 different markers for aggregated tau in CSF and cerebral spinal fluid of these patients. And what they were able to find is, as we moved from young patients all the way on the left of each of the graphs to cognitive impairment here, where it's just marked by CU with the plus, of a like prodromal Alzheimer's disease state, so they're likely developing toward Alzheimer's disease, all the way to a diagnosis of Alzheimer's disease, we see aggregated tau increase in the CSF. So we're able to show how these levels change over time in other diseases. So now it's going to be taking those same tests and using them for Parkinson's disease. And finally, what we're really able to do well for these patients now is look at PET scans, or positron emission tomographies. And what these are is you take a radioactive tracer that's specific for different proteins, or you can even do glucose to see how your brain uses the energy. This is infused into patients. It's, even though it is a radioactive, it's not harmful. And then we put the patient in a PET scan, which gives small levels of radiation that make the tracer glow. And because of the work in Alzheimer's disease, we have tracers that are available that measure aggregated tau. Currently, there are no tracers available to measure aggregated synuclein, although that's in the works. But because of how long we have been studying Alzheimer's disease and have understood the importance of tau in Alzheimer's disease, that field is more advanced in this realm. So when they use PET scans to look at patient brains, so you're going to see the patient brains shown in gray, and the areas of red to yellow show increasing signal from the tracer, showing increased amounts of aggregated tau protein in that place in the brain. And what we can look at just from the images and on the graph where they graph this, so you can see it really easy, is that as we move from Parkinson's disease patient that has mild cognitive impairment symptoms, we increase the amount of aggregated tau that we can detect in the brain, which is really quite remarkable. And this amount of aggregated tau seems to be even higher in the case of dementia with Lewy bodies, which has more significant cognitive decline and more cognitive symptoms than a normal Parkinson's disease patient would. And as we continue to develop these assays, this is going to be really useful for helping detect patients who may be at risk for developing mild cognitive impairment. And right now, in the short term, identifying patients at risk for mild cognitive impairment is about as good as it can get. And we can also use this to understand the mechanisms of how disease progresses. But why we care so much about this is in the long term, when we continue to develop new treatments, especially in context of dementia and tau aggregation, we could potentially slow or prevent the onset of cognitive decline in Parkinson's disease patients, right? And we also know there's an association between aggregated alpha-synuclein and the aggregated tau, and they seem to work together in some cases, like I showed you with the mitochondrial dysfunction hypothesis. So if we can target both proteins and get them to stop aggregating, we hope that may really make an impact in patients and really improve treatment options and potentially help stop disease progression overall. This is an active area of research with still a lot of studies and work that needs to be done here. And I would just like to say, we're very lucky here at the Van Andel Research Institute. The Van Andel family donated a large amount of money for us to create this West Michigan neurodegenerative disease program, or it's called the MIND program. And this allows us to work with clinicians at local hospitals to receive blood, CSF, and urine from Parkinson's disease patients. Well, it's all sort of a variety of neurodegenerative diseases and healthy controls, but in the case of this, it's really exciting for us to look at Parkinson's disease patients and use these samples to study this, to create biomarkers and understand how Parkinson's disease may be genetically linked, specifically here in West Michigan. And we also are extremely lucky to have a brain biobank here that was able to be started, where patients who selflessly decide to donate their brains to research upon their passing are able to have their brains sent to us, and we're able to use them for studying Parkinson's disease and understand how these proteins aggregate, where they aggregate or clump up, and what aggregated proteins seem to appear together. So we can better our research, our models, and affect treatment. And if any of you are interested in learning more about this, it is on our Van Andel website, and you can contact Erin Williams, who is in the back of the room also. Erin, if you'd like to say so people are. I am pretty far separated from this, but Erin does all of this work and coordinates with everyone. Yes.
I donated for last year. Would it be going to this, or should I do it again this year?
I don't believe Erin or the MIND program is speaking this year.
No, we're not. We don't have a slot this year.
No, they don't have a slot this year, but if you find Erin or email her, she is happy to talk more about this program to you individually. And just finally, I would like to thank Cure Parkinson's Foundation, Van Andel, all of our incredible organizers for this, the co-chairs of the Grand Challenges Conference, Helen and Rosie here, for rallying to the challenge, our planning committee, and then finally, most importantly, all of you and all of the Parkinson's disease patients and families that continue to be active with the research we do here and being interactive and giving samples of biofluid or even brains, because you guys really are the reason we do what we do, and all of you that are here excited about the research, wanting to learn about the research, help drive us forward.
Alysa, thank you very much indeed. That was really an important talk to understand some of the work that's going on here at the Andel Institute and looking at how different proteins are working together in some people with Parkinson's. I also do want to add that this is the first time that Alysa has spoken to a group of people living with Parkinson's. And so actually, I think full credit is the level of communication right is a real challenge. So thank you very, very much indeed. Has anyone got any questions for Alysa?
Yes.
Matt?
Yes, about detection of tau in plasma versus detection of tau in CSF.
Yes.
You mentioned that it seems like there's some success in detecting aggregated tau in plasma, less studied and perhaps less successful in CSF. Though in the field of Alzheimer's, quite successful in CSF. And I'm also more, I think, wondering, there's an explanation of how on earth aggregated tau is ending up in the plasma and the blood, given blood-brain barrier, to be able to detect it in plasma versus CSF, where you would assume that given that's surrounding the brain, it would be more present.
Yeah, so that's a really great question. And we do think the brain is fairly protected, but especially in disease and inflammation, that protection barrier decreases. So there's more crosstalk, I guess, between the outside of the brain, called the periphery, and the brain itself. So, and it's also important to note that a lot of these mechanisms that I talked about with inflammation and mitochondria in the context of neurons also do occur in other cells in the body, right? Other cells express tau because they're important for structure of every cell in your body, and other cells express alpha-synuclein. So these can occur really anywhere in the body, and we really don't fully understand why we can measure it. We just know we can, and we like that we can. Yeah.
My sister had TBI about in August. She had an aneurysm. She get more likely to have Parkinson's because of that.
I'm really not sure. There aren't a lot of great associations between traumatic brain injuries and neurodegenerative diseases. In some cases, we think it might help, but it's really repeated, the chronic states, the repeated, it's like, I think we might all be familiar with the link between like head hits in football and CTE. It's a similar concept that it's the chronic state, it's the repeated state that's going to be really important for developing neurodegenerative diseases.
Question at the back.
The tau is a protein, and when it's breaking down or clustering together, does that ever? How long does it take to do that, to cluster?
It depends. In the body, it's really hard to know, but in our simplified systems, like in that neuron growth that I talked about, we've also added just tau fibrils, and we see pretty robust or a lot of seeding into those cells within 3 weeks.
Unclustered, is it completely separated, or once it's clustered, is it over?
So when it's not necessarily over, I think, right, if this happens or a few of the tau proteins become over modified, the cell has a way to deal with that the same way it does with alpha-synuclein. So a few misfolded proteins isn't going to lead to neurodegeneration, but it's the cell not being able to handle that, or too much of those proteins happening too fast, that leads to the large clumping up.
Right.
So there are pathways to deal with it, but sometimes it can't occur fast enough, or the clumping up happens at a rate that's too fast for the cell to deal with.
It'll turn a vicious cycle, then it'll go to inflammatory response.
Yeah.
And then it'll cycle around with.
Yeah.
Okay, thank you.
Fantastic. Thank you very much indeed. I think it's